Good morning. This is Dr. Ankita Gupta, Assistant Professor from Department of Microbiology, and I am here today to teach about culture media. So culture media, they play a very important part in our microbiology. So the topics which I will be covering in this presentation would be what is culture media, what is agar, then types of culture media. There are several types of culture media like enriched, selective media. What is the difference between enriched and enrichment? How do you sterilize media? Then what are the anaerobic culture methods? We all know that bacteria are both aerobic and anaerobic, and for these separate criteria of bacteria, there are separate kind of culture media. So moving with our presentation, first of all we should know what is culture. So culture is defined as the process of cultivation of microorganisms in the lab for the purpose of studying them. See, microorganisms, as you all know, microbiology is the science of microorganisms, and we have to cultivate it in vitro, that is in our lab, so that we can study them. What we have to study? We have to isolate them. We have to isolate and identify them. Which type of bacteria is this? Because the treatment, the empirical treatment of the clinicians, it depends on identifying the microorganisms. Then we should also know what is medium. So medium is a combination of ingredients that will support the growth and cultivation of microorganisms outside their natural habitats. What are the natural habitat of bacteria? Any living host. Can be a human, can be an animal, but when we will grow these microorganisms in vitro, that is in lab, we require a medium, and a medium is a combination of ingredients. Okay. Why we need culture media? So as I have told you earlier also, to identify the bacterial species in patient samples. As we are dealing with samples from patient, we have to identify the bacterial species that cause infection in patients. We have to isolate these bacterial species. So the need is to isolate the bacterial species. Then we have to also detect its antibiotic sensitivity. To which all drugs is the microorganisms sensitive or resistant, and it will all be helpful for the clinician to start a treatment protocol and will in turn lead to help of patient. So I'll just be giving a very rough idea how the culture is done. We all know that patient sample comes in a universal container. So we obtain the patient sample. There can be several samples like urine sample, pus sample, blood sample. It is all brought into a laboratory in which there is a requisition of culturing, of culture and AST reporting. Then from that sample, from the universal container, we inoculate the sample to a culture media, and once we inoculate it into a culture media, we give it time to grow. So, and the time at a specific temperature. What temperature? As we all know that human body temperature is around 37 degrees Celsius. So we have to provide the bacteria 37 degrees Celsius in vitro also. For that we have incubators. So in incubator we allow them to grow at 37 degrees Celsius overnight. And most of the bacteria, leaving some, there are some exceptions, but most of the bacteria, they grow within 18 to 24 hours. And most probably by 48 hours they grow. Then once they are grown, we have to identify them, and we have to also put their antibiotic sensitivity testing. Now moving on to classification. How are the culture media classified? So culture media can be classified under various categories. Now depending on the physical state, you all know the physical state what we have liquid. In microbiology we also have semi-solid and then solid. So according to the physical state, culture media can be divided into either liquid, semi-solid or solid media. The example of liquid media will be peptone water, nutrient broth, BHI. We will discuss them one by one. In semi-solid, we have nutrient agar stabs, and then we also have an OF media. For solid, we have blood agar, chocolate agar, LG media, etc. So this is the categorization according to their physical state. Then we can also classify the culture media according to oxygen requirement. So if used for an aerobic bacteria, we require an aerobic medium, and for anaerobic bacteria, we will require an anaerobic medium. For example, Robertson cooked meat broth. Third categorization is according to the special purposes. Okay, so first we have a simplest media, that is also known as a basal media. So simplest media promotes the growth of simplest bacteria that can grow easily anywhere. Okay, then we also have complex media. Complex media includes enriched media, enrichment media, selective media, indicator or differential media. In complex media we are adding something extra, okay, for some special type of bacteria. Then we also have a transport media. Transport media means whenever the lab is away from the place of sample collection, as if the sample is collected in some village or at periphery, and from there we have to transport it to some lab. So we take the sample in those transport media. In transport media the bacteria does not die, it remains viable. So that was the categories according to the that were the classifications of culture media. Now moving on to constituents of media. What does a culture media consist of? So most importantly it consists of water, okay, then electrolytes. As electrolytes they are very important for the growth of bacteria, and it also has peptone. So these are the three things that are present in all media. What is peptone? Peptone is a mixture of partially digested proteins. So these three things are universal in all media: water, electrolytes and peptone. If we add agar to it, okay, agar is obtained from seaweed and it has no nutrition of its own. It is only a solidifying agent. If we add agar to water, electrolytes and peptone, we'll get solid media. Okay, then we can also add meat extract, serum or egg. There are some bacteria that grow well in egg, so we can also add egg. So in liquid media there is no agar, there is water, electrolytes and peptone. Okay, and in solid media there we add agar also. Agar, again I'm telling you, it is obtained from seaweed and it does not have any nutrition of its own. Percentage of agar used in solid media is around 2%. Then there is also semi-solid media. In semi-solid media we used around 0.2 to 0.5% of agar. Then there is one hard agar also. Hard agar, it is used, the approximate concentration of agar here is around 6%, and it is used in special cases. It is used for Proteus species to inhibit the swarming in Proteus. So just a rough idea of how the culture media is prepared. So for preparing culture media we have this dehydrated media that is in the form of powder that we get commercially, and once that powder is obtained we just mix that powder with water. There is a specific capacity of that powder to be used. We mix it with water. Distilled water has to be used, that should be sterile, and then we will go for sterilization of that thing. It is all mixed in a beaker, and then this beaker is put into an autoclave. So autoclave, it is a machine for sterilization. You can see it in all labs. So autoclave, we sterilize the media. Once the media is sterilized at specific temperature and pressure, it is poured into petri dish. You all have seen petri dish. These are round dishes with a cover on top also. It can be made of glass that can be reused, and they are also made of plastic. Those plastic ones are disposable. So we'll pour that liquid media that has been sterilized in an